Good afternoon, good evening, and good morning at various places across the world. And I'm very glad to be part of this interesting session. This is the first time I'm talking at Global Ayurveda Forum because I am not really an Ayurveda practitioner. I'm a basic scientist and have been working on the basic science all of my career. And I think this is the right place for me to express my thoughts and all the work that we have done from Patanjali Research Institute, Haridwar, India. So on the foremost, I would like to express my gratitude towards the organizers of this meeting, the coordinator, Dr. Rahul, and the session chairs, Dr. Sanket, Dr. Saket, and Dr. Manoj Shankar Narayana. So my humble gratitudes and namaste, om, and we will now start with the science. So I do have a presentation and I'll just try to have it shared with you so that I can go through it. And I hope I have close to 40 minutes time as the schedule suggests, and I'll try to finish within that time and try to take you through the journey that what Patanjali research has been through the medicines of Ayurveda against the present pandemic, COVID-19. So can you see my slides, I hope?
Yeah, we can see. Yeah, Rahul.
Is it visible to all of you? Can I get a confirmation that my slides are visible to you? Hello?
Yes, it is visible. Dr. Anurag, your slides are visible.
Thank you very much indeed. So the title that I'm going to talk today is a Coronil Ayurvedic Intervention for COVID-19. And as I introduce myself, my name is Anurag Varshney. I'm PhD and MBA. My PhD is in molecular neuroscience and MBA in international business. I am the Vice President and Head of Drug Discovery and Development at Patanjali Research Institute, Haridwar, India. And my contact information at the bottom of the slide, which is my email ID, anurag@prft.co.in. So what I want to show at the first and foremost, that what is the aim of the institute that I'm coming from. We are the lineage partners, we are the lineage carrier of ancient Ayurvedic wisdoms at Patanjali, and our holistic aim is to improve the health and the quality of life of people. And at Patanjali Research Institute, we want to achieve it through research and innovation. So just I want to take a couple of my slides just to introduce about my institute because this is fairly new. This was inaugurated in 2017 by our Honorable Prime Minister of India, Shri Narenda Modiji. It's a 4-year-old institute now. This houses various research laboratories, and we do cutting-edge research at this laboratory. And there are few glimpses of the time of inauguration in 2017 by our Prime Minister, Shri Narendra Modiji. Our forefathers, our guides, and our source of inspiration, Swami Ramdev Ji and Acharya Balkrishna Ji, are also shown in the same images. Now, the next slide shows our vision. What we wanted to do at this research institute, because most of the time when we talk about research in Ayurveda, it has been seen in different parameters. So what we intend to do, we intend to actually look at the Ayurvedic and herbal information in the public domain. Sometimes it is highly unstructured, sometimes it is very structured. And then we also want to take a rich history of medicinal plants and pass through both of these through the funnel of scientific methods. Those scientific method funnel would have a several components, for example, old wisdoms, modern practice, larger scope of delivery, multi-pronged approach, and try to reach to a research Ayurveda. And what we tend to achieve and what's our target and what's our intention have been to have an Ayurvedic medicine which is very well characterized in terms of the research. And in that process, we would then scientifically validate an evidence-based medical system, what we call Ayurveda now. So then next slide shows our strategy, how we intend to do this. So when I look at the strategy of the whole institute for last 4 years, I could see that we can set it up on a set of a pyramid, and pyramid having 4 major stages: initiation, transition, actualization, and excellence. So we whole work in almost like 4 to 5 years before, where we had the visionary leadership, ancient wisdom, and a pharmacy to support the drugs that we're going to work upon. We transit through the noble processes and try to go towards patented medicine and set up a world-class infrastructure to do advanced research on Ayurveda, almost in the same way one would have for other pharma industry laboratories. We actually set it up for the Ayurveda here in Haridwar, in Uttarakhand, in India. And then we move toward actualization. And in my humble opinion, we are right now at this stage for our center. We right now have an energized workforce. Close to 300 scientists work here with several PhDs and postdocs. We get ignited thoughts. We really think very deep about the science behind the drugs we're talking about, and we try to deliver the quality. That means we try to generate things which would be acceptable throughout the world. And in the end, we are hoping to reach the process of our excellence. The pinnacle of excellence that we are trying to reach is the universal acceptance of Ayurveda. So next slide shows the research strength and the accreditations that we have at Patanjali. So I really chose to show this slide here because that actually talks about the capability we have in terms of the research at Patanjali Research Institute. So our labs are having 3 laboratories which have NABL accreditations with a dedicated QA compliance. NABL is a quality parameter in India to actually see the laboratory accreditations for a defined delivery. We also have DSIR accreditation from SIRO, which is again Indian government accreditation for the research capability of an institute. As I said, we have more than 300 scientists, several PhDs and postdocs with the global experiences that we are working together day in and day out here at Patanjali Research Institute. We have a full-fledged registered animal house as per the government norms for conducting animal experimentations, which is governed by Institutional Animal Ethics Committee. So this is one of the rare things for the Ayurvedic research, because to generate a proof of principle, sometimes we have to go through a reverse pharmacology approach, and we chose to go that path and try to see the efficacies or at least a pharmacological effect of a drug in a disease model. And for that, we require a dedicated animal facility. So we chose to set it up here, and we have that in a fantastic way. Right now, we are in the process of upgrading that facility to take the GLP accreditation for the same. Our research laboratories are in compliance with Biosafety Level 2 and is governed by Institutional Biosafety Committee. We are right now in the process of setting that up to Biosafety Level 3 so that we can handle the viruses as well. Of course, both of these facilities, the biological research lab and the animal house, have a dedicated clean and service corridors to make sure that there are no mix-ups. In addition, we have an Institutional Ethics Committee for human clinical trials, and most of our trials are registered in the CTRI, which is Clinical Trial Registry of India. And last but not the least, which is the fantastic support we have, that at Patanjali, we have an 180-bed Ayurvedic hospital with NABH accreditation, which becomes our source for conducting a lot of basic research. We have an approved Ayurvedic medical college with a post-graduation degree approval in 8 subjects. On our roster in Patanjali across India, we have 1,500 medical doctors who are working on our Chikitsalayas, that means the small OPD centers across India, and the daily footfall across India for those hospitals, those clinical centers are 15,000 patients per day. And we acquire and compile the data, the primary data from these patients in our electronic medical record sheet. And at this moment, we are close to 70 lakhs, close to 7 million patient data that is available with us at this moment. So what I would like to show in this busy slide and try to convince you that we do have a diversified research framework. And with that background, we start doing our research. So the next slide, if you see on your screen, that talks about the major segments that we have at Patanjali Research. So the basic thing that we do a fantastic herbal research, which you see on the left-hand side, the yellow circle, where the team of close to 150 scientists are actually compiling all the herbal information available across the world, not only India. So like Ayurveda, there are 61 other type of herbal medicinal systems which are based on herbs. So we are compiling all the plants which might have or which have been reported to have a medicinal property across the world. Total 65,000 plants have been now compiled. Their information and be done in 16 different languages. And that's a mammoth task that Patanjali Research Institute is doing and generating something called World Herbal Encyclopedia, which would have close to 100 volumes. The first 2 volumes are out, and we are in the process of getting those up too. The second and foremost component of Patanjali research is the drug discovery of development, where we actually follow the norms, the rules, and the protocols, what one has to do for the pharma industry too. It's just that we wanted to develop the proofs, evidences, and scientific supports for the drug's functionality in a way, in a manner that people outside India are also able to understand, appreciate, and agree with it. So for that mandate, the Drug Discovery and Development Center has been set up at Patanjali Research Institute. We have paintings, plant paintings, and herbarium, which are registered. We have close to 65,000 canvas painting of different plants and their line drawings, which also becomes a fantastic museum for the researchers to follow and try to understand what they want to do for their research. There's a dedicated accredited herbarium at Patanjali Research Institute. We also work to preserve the ancient documents which have been written by our saints, and those are being scanned, compiled, and generated in form of books. So far, 26 crore to close to 2.6, well, not 2.6, 0.26 billion pages have been now scanned and have been preserved by doing this activity. And these ancient documents are like several centuries old, and we have got hold of through across India expeditions for looking for those documents. And we have a dedicated team which looks at the yoga research, which is led by Dr. Sully Tallis, which does the specific research on the yoga, which again comes in the purview of Patanjali Research Foundation. When we do this work, we also get support from other teams across Patanjali, which is a big conglomerate. We have a Patanjali Organic Research Institute, Central Labs at Patanjali Ayurveda Limited, New Product Development Team in the Patanjali Ayurveda Limited, and Patanjali Hospital and Medical Colleges. So in total, we get a whole bouquet of the services, capabilities, and testing that we can do on Ayurvedic medicines. So in my next slide, I just want to show in the drug discovery and development, the basic capability we have. Our whole research capabilities are divided in 4 major sections: chemistry, microbiology, in vitro biology, and in vivo biology. And these all 4 functions collaborate very deeply with each other and trying to actually resolve the question that we are looking for. In the same slide, there is a glimpse of the infrastructure that we have in our laboratories. As you could see, there is an LCMS machine, there is a GCMS, there is a whole set of HPLCs and the other biologically relevant equipment too. From animal house, you could see the individually vented cages on the bottom left photograph, and on the right, you see our in vitro biology labs. So in total, we are geared up, and we are very happy to report that we have been conducting state-of-the-art research for the Ayurvedic medicines and trying to develop new formulation too. So this is the basic summary so far for last 2.5 years at Patanjali Research Institute, where we have a disease-driven approach for Ayurvedic drug discovery. And these are the therapeutic areas we have been focused on: inflammation, metabolic disorder, neuroscience, and infectious diseases. And the numbers on the right-hand side of these bars tells you the number of medicines that we have worked or developed and which have been now in the pipeline for the release. So in total, close to 100 different articles, 100 different test items, which have been tested and validated at Patanjali Research Institute. So with this introduction, I come to the core of my presentation. And in the next 18 to 20 slides, I will take you through Coronil. And I'm sure most of the people in India and across would have heard about this medication as well. So this is the medication, this is the Ayurvedic drug, which has been generated by Patanjali Research Institute for the pandemic we are into, COVID-19. And this is a supporting measure, approved medicine for COVID-19 by the regulatory authorities. So in my slides, I will not go into those regulatory components, but as being a scientist, I will try to take you through the science behind it, how we developed, what we did, what was the path we followed to do, what kind of data we generated, and where did we publish all of it. In the end, I will try to convince you that there is a real science behind Coronil and its genesis. And of course, I would really heartily welcome your comments, your queries, and the suggestions once I'm done through that. So let's take this ride called Coronil. So this slide tells about the Ayurveda battle against COVID-19 pandemic. And I'm sure most of you might know already, and you know by their heart, that COVID-19 symptoms, which you see on the left-hand side, all of these things that happen, there's a fever, there's a cough, there is a lot of other things that also happen. And how does this disease occur? That most of you now know that COVID-19 is caused by a virus, SARS-CoV-2, and the target organ for this virus, the primary target is the lungs. So the infection happens through the nasopharynx and goes into deep lungs, and a lot of things happen. So over there, cytokine storms happen, and the lung capacity to breathe, to ventilate decreases, and a lot of things, good and bad, happens at that time as well, which lead to most of the fatality that we're talking about and that we have read about it. In addition to this disease, there are other than the human health, it has an effect on the psychology and also on the society. People develop mass hysteria at that time. There's anxiety, there's a depression, there's a stigma, there's a stress, and it also affects the economics of the society. There's a loss of jobs, disrupted workplaces, disrupted markets, and fall in GDP. A lot of people start to learn to live life differently during this COVID period. So then what Ayurveda can do to take care of some of those. So for there, Patanjali start working on it, and the battle of Patanjali to fight against COVID-19 came out in form of the Coronil. Coronil came out in form of a kit, which has a Coronil tablet, Swasaribati tablet, and Anutela as a nasal therapy. Today, I will talk exclusively on the Coronil tablets. Am I going too fast, or is it okay?
It's okay, Mr. Varshney.
That's fine, or am I speaking too fast?
No, it's fine. Please go ahead.
That's fine. Thank you very much indeed. So this slide shows the capability that we had, the planning that we started with. So in February 2020, when COVID-19 was at its prime in China, and there was no specific reporting in India, we started to think that maybe there's something, sometime in future we would have to deal with it. And so happened that we have to deal with it in March itself. And we start to think that from the rich history of Ayurveda, what one could do, what is the information that is there out there in our ancient text, which we can try. So we then start looking through the textual information, and there was not much available simply because the COVID is a disease which was not reported at that time. Fairly simple. So what we can do about it? So then we turn towards our computational modelers, our friends who do a fantastic computational modeling. And in February itself, we screened close to 1,200 phytochemicals present in close to 150 different plants. All of those plants which have been reported in Ayurveda to have some significant effect against fever, against infections, against and just to enhance immunity as well. And what we found, the 3 plants which could be usable. One is Vidania somnifera, ashwagandha. Second is Tinospora cardifolia, giloy, and third is Ocimum sanctum, tulsi. And within these 3 plants, what we could also find the specific phytochemical which is present in those which might be critical. So what we try to do, that which part of this disease we can look into. So you remember this COVID virus actually interacts with our human cells through its own receptor, and that's called spike protein, which is outside on the periphery of the virus, which interacts with a specific receptor in our cells, and that receptor is called ACE2, angiotensin converting enzyme 2. So what we did some computational simulation where we got the receptor binding domain of spike protein as well as the ACE2 protein of human, and try to see if there is anything we can dock into or simulate into in terms of time which might affect this binding. And we calculate the binding energy and its modulation by the compounds that we are looking at. So we found a compound called vithinone from Vidania somnifera and a compound called tinocorticide from Tinospora cardifolia, which could actually does exactly the same what we are looking for. Both of these compounds individually decrease the binding energy of SARS-CoV-2 spike protein and that we could simulate and found that this was actually stable inhibition and which could lead to some pharmacological effect. This we then went ahead and conducted lots of biochemistry, cell biology, and animal experimentation, which I'm going to take you in the next slides later. But I want to also mention that from a plant called Tulsi, Ocimum sanctum, we found a compound called sculptorine, which found to bind to a target within the virus, and the target was dependent RNA polymerase. So remember, SARS-CoV-2 virus is an RNA virus, and it requires RNA to be replicated. And that is done by a protein called RNA-dependent RNA polymerase, RDRP. And we found that compound from Tulsi was actually binding at the active site of that enzyme and was competing with the ATP, which would mean that when this compound is present, then the replication of the virus would be hindered. We have published all of these findings in various journals. The citation there at the bottom of this slide. One which is about Vidanon, which came in press like day before yesterday. It was accepted in January in a journal called Drug Design, Development and Therapy, where we could show that Vidanon does indeed attenuate SARS-CoV-2 and ACE2 receptor interaction. The one about tinocorticide have been published in a journal called Computational Chemistry and High Throughput Screening last month, which is also there in the public domain. And what we're very happy to see, once we start reporting these kind of things, the other groups in India also start to follow the same kind of experimentations. And we also found a few compounds from ashwagandha, Vidanon and Vidaferin A have been reported from other groups across India, which shows a similar kind of finding that we are looking at. So we got a third-party validation and we feel very excited about it. And really, we are thankful for their contribution towards moving in the right direction. So the next slide. This shows the mode of action, which I'm sure most of you know this. But what I want you to do is to focus your attention on the right-hand side of the slide. I'll try to get a marker if I can. Yes, I can get a pointer here. So this part, can you see my pointer? So this is actually the interacting partner of these two proteins, ACE2 and RBD. ACE2 is the human protein, RBD is a viral protein, and this is actually the Vidanon or tinocorticide sits in this pocket, and that pocket it then becomes energetically unstable, and then the interaction between these two proteins would be actually broken down. But that's again speculation or suggestion that come from the in silico modeling. For a biologist like me, we need to see, does it really happen in the experimental conditions? So then what we chose to do, we actually then went towards our biochemistry, and we did a basic biochemistry where we got both of these protein purified and as a single binding species, and then we connected both of them through an ELISA and see if we could introduce Vidanon or tinocorticide and see if that interaction can be broken by doing those simple biochemical interaction experiment. So the graph on the bottom of this slide, which you see where my cursor is there with the laser pointer, this is actually the percent inhibition data of that interaction by Vidanon. And what I'm very happy to report, the IC50 of that inhibition came out to be 0.3 nanogram per ml. Yes, 0.3 nanogram per ml, which was actually very, very, very low. That suggested us that this would be a very specific interaction too. So the biochemistry support what we really saw in our computational modeling. So what next? Then the next thing, can we do some cell biology? For that, we would have to set up an ELISA-based assay for these kind of interaction. This is just a schematic. And what we also did, that we took ashwagandha, which is Vidania somnifera, Tinospora cardifolia, and Ocimum sanctum, and put together all three of them in a fixed ratio and developed a formulation, a tablet, which you could try later in the clinical trials called Coronil. And that was the first genesis. We started to look that, okay, maybe this thing might work it out because these would have those compounds that we are looking into. So going forward, I will refer all these three plants or I will refer Coronil as such. So the next slide shows this data that I was talking about. So this is a cell biological validation. So what are we doing it here? Well, we actually looked for, well, this is not a cell biology, so my apologies. This is again a biochemical validation where we looked for the activity of the coronil. So the left slide on left graph on a slide where here you're seeing it, this was the wild type spike protein that we could get and interaction with the ACE2. And if you put a different concentration, 0.001 microgram per ml all the way up to 30 microgram per ml, this you see a % inhibition of the interaction that goes up significantly. So we could see a very nice dose-dependent inhibition of SARS-CoV-2 spike protein and H2 by biochemical measurement. Now, at that time, we also got to find there were 2 different mutants of this virus which were out there. One of them was D614G and second was W436R. This is the D614G and this is W436R. So we also got those mutated spike protein and also try to see whether this interaction can also be modulated by Coronil. And we're very happy to report that yes, indeed, Coronil could actually inhibit the mutant protein interaction as well. That means the clinically relevant mutation of the SARS-CoV-2 spike protein could also be inhibited of its interaction with the human protein. For the second mutant, we even saw even much stronger response at almost all the doses tested. There is a clear dose-dependent response, but we also found that yes, it does happen. So from the biochemistry, we got excited that, oh, yes, we are in the right track. Now let's take it further and do some cell biological measurement. So here this comes a similar kind of data, but here we chose to use the human cells. So we got human lung cells we call A549 cells, which are the carcinomic cells from human alveolar epithelium. And those cells then we actually treated with the 3 mutants that we have for SARS-CoV-2, wild type for 436R and 614G. And those then we actually looked, can we also measure the cytokine response which getting elicited? So now we know, all of us know the cytokines release or over elevated level of cytokines in human patients becomes a critical factor for the disease prognosis. So we chose to measure that first, that if you put a spike protein from the virus on the top of the human cells, does this induces cytokines? If it does, can you control that by putting Coronil at different concentration? And we chose to look at the 3 specific cytokines, IL-1 beta, IL-6, and TNF-alpha. These 3 have been the predominantly specific cytokines for these kind of functionality, IL-6 being the kingpin. Then we tested now basically 9 experiments. So you have 3 different mutants and 3 different cytokines on the same cell lines. So the graphs at the bottom, what you're seeing it, this is for the wild type. This is for the wild type, IL-6. This is for one mutant where the IL-6 was not even induced, which is reported as well in the clinical setting, that this mutant does not induce much of the cytokine release in the human patients, while this wild type does it. And the different doses of Coronil could reduce it in a concentration dependent manner and statistically significant manner. Again, same thing for IL-6, you look for the other mutant and we could see it reduction. This is for TNF-alpha. Again, this mutant did not have any response, while other 2 did have it, and we could see a very nice dose-dependent inhibition of that cytokine release of TNF-alpha. Similarly, IL-1 beta. So we got to see, okay, other than the interaction, Coronil is also controlling the cytokine release. Now, does this mean it is having an effect on cytokine processing per se, or it's affecting the virus entering at all towards the cell? Because these are the spike proteins which would be the first anchoring points. So my next slide shows the experiment we really want to work upon. So this is a slightly busy slide and I'll try to go through a bit slowly here. So since at this we were doing this at the time of April or May 2020, where the COVID menace was at its peak, and we could not get hold of the virus simply because it required BSL-3 facility, biosafety level 3 facility, which we didn't have at that moment. And again, getting a viral strain was also risky for the experimenter, the scientists that we have over here. So what we chose to do, we actually had a fantastic support from Professor Steve Pohlmann from Göttingen, Germany. He was super kind and very helpful to help us generate something called pseudovirus. What pseudovirus is? So pseudovirus is actually some other virus, which is non-infective in nature, but has a capability to replicate. And what we did that we coated that virus with the spike protein of the SARS-CoV-2 virus. So what happens that now we have a species which has an exterior feature like SARS-CoV-2, but doesn't have an infectivity like SARS-CoV-2. That would mean that that becomes safe to do the experiment with. Our scientists would not be at risk of using it, yet we could actually decipher whether this drug that we have in our hand or any other drug that we are working with is showing any functionality of the spike protein interaction with the human cells, because now they're in the virus. So we did one more thing. What we also did, once we make the recombinant virus, we packed that virus with a luminescence gene inside it. So luminescence becomes a marker. So basically, if the virus exists, we can measure the luminescence in the solution or in a cell. And if the luminescence is coming there significantly, that would imply that virus is present. So in place of counting the virus, we chose to go for the readout. So that was a recombinant method to track the virus and also to measure whether this is going to go inside or not. So that comes to the specific query that I had in the last slide, whether the coronil affects the entry of the virus in the human cells or not. And the answer is, it does affect the entry. And you look at the data at the bottom where I'm getting my cursor on, those brown, red, pink bars, this is actually showing the percent inhibition of the viral entry inside the cell. This is the normal cells, these are the one with the positive control, and these three are the doses of coronil. So when you look at the coronil from 1, 3, and 10, the total amount of luminescence coming from the cells which are being infected with the pseudovirus get decreased in a dose-dependent manner. What that implies? It implies that yes, coronil can actually affect the entry of the virus inside the human healthy cells. And now, does it also mean that what we saw in the biochemical measurement, does it also affect the cytokines? And then we went ahead and did ELISA for same three friendly cytokines we have been working with, IL-6, IL-1 beta, and TNF-alpha. And there we could show that cytokines which get released by the pseudovirus on human healthy lung cells, it also get attenuated in a dose-dependent manner by coronil. The line in the blue are the different concentrations. The bar in the blue are in different concentration, which get inhibited by coronil. So we have IL-6, TNF-alpha, and IL-1 beta. So to take it from here, that shows that coronil indeed could rescue the cells from a cytokine storm. And of course, it prevented the viral entry inside the cell. So this has been again published in Journal of Inflammation Research, and well, it is coming out day after tomorrow. It has been accepted for publication last month. So we just finished the galley proofing and it's out there too. From in vitro biology, we have been doing the experiment. Then comes the next thing, that what we can do. Because when we talk about cytokine storms in the cells or in the human, generally it's the one cytokine release that causes other cytokine to release. So we thought, how about we do the same thing in our laboratory? So we went ahead and took again A549 cells, and this time hit those cells with IL-1 beta. So we had a purified human IL-1 beta. We can put on the top of those cells and look for the IL-6 and TNF-alpha release, because it's known that IL-1 beta itself can cause IL-6 and TNF-alpha to release, and that's how the whole storm thing happened, because that is the crosstalk between the different cytokines. So if you look at these bar charts on the left-hand side here, this is IL-6 and this is IL-1 beta. In a normal control, there is no significant value of IL-6 in the supernatant of the cells. But when we hit with the IL-1 beta, which is 500 picogram per ml, we see a sizable, close to 1000 picogram per ml of IL-6 present in the cell culture media. And when we treat these cells, pre-treat these cells with coronil, we see a very nice dose-dependent decrease at different concentrations. In vitro level, these kind of findings are very significant. We also went ahead and looked for TNF-alpha in the same experiment when we hit them with IL-1 beta. And again, TNF-alpha shows a much stronger, much robust response for the decreased IL-1 beta-induced TNF-alpha release. We didn't stop it there. We thought if these two are critical, there has to be something downstream. And the downstream, it's actually a signaling cascade. So there are signaling domains, there are different transcriptional factors which control the release of these cytokines. And one of the critical one is NF-kappa B. So NF-kappa B also plays a significant role. So we had a specific cell line, we had a specific reporter gene cell line, where if we hit those cells with TNF-alpha, the NF-kappa B level goes up. And that we could detect by doing a reporter gene assay. So we employed those technology available in our laboratory. And at the bottom bar chart, you can see these are the normal control and these are the TNF alpha hit cells. And you can see there is this very sizable increase activity of NF kappa B AP1. And again, when these cells are pre-treated with Coronil in a dose-dependent manner, we could see a significant inhibition. So that makes us believe that yes, Coronil is working at that lead as well. Am I going okay or too fast?
It's fine, Mr. Varshney. Please go ahead.
That's fine?
Yeah.
That's fantastic. So now comes the very critical component most of people talk in Ayurveda. The bigger question that we get from a drug discovery friends of mine, since I come from a drug discovery, the classical pharma industry drug discovery, that what are the chemical signature a drug would have? Can you quantify, qualify, and make sure that you get everything the same what you're looking for? So for me, that was very challenging for myself as such. So what I did, then I went ahead and talked to my chemistry friend. My analytical chemistry friends were very happy to help. So we went ahead and did 3 different validation of the Coronil tablet. The first is LC-MS QTOF, liquid chromatography mass spectroscopy, where we could actually test these tablets and could detect the amount of compounds present in them. What you see on your screen is a spectra which come in the positive and negative mode, and the boxes on your left and right list out all those compounds that could be detected by LC-MS QTOF. So that tells you close to 44 phytocompounds was present in this tablet, which we could detect through LC-MS QTOF. From there, then we went ahead and looked for the validation that these compounds are there. Can we actually, again, identify and quantify by using other technology? So we went for HPLC, high performance liquid chromatography, and then we look for these compounds which should be present over there. We found those compounds: cardiofolioside, magniflorin, rosmaric acid, palmitic acid, vithinone, vithinosides, bidaferrin A, and bithanoate 4 as well, along with betonic acid and ursolic acid at different wavelengths. So the graph right in front of you, what you see, the pink color lines are the standards. That means we got a purified compound in single isolation from commercial sources, and we run in parallel with the extract or with the solution made from the Coronil, which is there in the blue line. So if you see the standard compound mix in the pink or violet, while the blue is the Coronil, and you can see those compounds that we're trying to identify were there in the significant amount. Since we knew the amount of the compound present in standards, we could actually then correlate back and calculate the total amount of compound present in the tablets of Coronil, and which were coming out pretty sizable as well. And then we didn't even stop there. We got 50 different batches of Coronil, which were manufactured by Dibbe Pharmacy, and tested this and tried to found that what is the level of variance we are seeing it. And we're very happy to report there were a very minimal variance. There were, of course, few compounds which are going up and down, but which does happen because of seasonal variance of the raw material that we procure from. But the 6 compounds that we set up as our primary markers, those were largely conserved throughout. So that gives us a feeling, okay, we are on the right track of manufacturing. We are also looking at the compounds at the right areas, and our tablets containing all of it, what we look for. But my chemistry guys was not even satisfied. He said, sir, if we have done LC-MS, we have done HPLC, how about we go for one more validation? Let's do HPTLC. So we then went ahead and did a HPTLC fingerprinting of the compounds present over there. So what you see on the top, those 4 colorful bars there and those black marks there, those are the single compounds present in there. For example, this one is vidaferrin A, this band, this is the rosmaric acid, this band. These 2 are the bands, the lane coming from Coronil. And as you can see, whether these bands are present there, whether these bands are present there. Similarly, if you go for a different white light derivatization, you start to export them better, and then you can quantify by looking for these peaks. So this is vidaferrin A, which matches very well with this peak. Rosmaric acid matches very well with this peak. Ursolic acid matches very well with this peak. Similarly, things happen for other components too. And so we were happy that by using 2 different validation technologies, we could find a similar findings. And again, different batches was tested, and pretty much conservations were observed throughout the batches we are looking at. So from here, the biologist inside me got convinced that we have a very stable formulation that we are working on. So the biological response that we would see in our cell lines or in our biochemical experiment or even in our animal experiments, we would be able to actually very comfortable about it, that we are having the right starting material with the least amount of variance possible. So next slide. That tells me something very exciting. So I'll take a sip of the water and then I go into it. So this is again a very hardcore in vivo biology. Now, why I want you to take your attention on this slide. The basic thing that the difficulty that happened for a SARS-CoV-2 virus in vivo experiment, that the classical animal models, which are rat and mice, are literally immune for SARS-CoV-2. That means that this virus does not infect those animals. So if it's not being infected, you cannot see the propagation of the virus or any effect of that virus in those animals. So what you do, either you wait for mutant animals to come, all those transgenics and knock-ins and knockouts, which takes its own sweet time. Still the work is going on and a lot of companies are working on those to generate those animals. Or second option we had, that we try to apply our thoughts on something else. And then one of our colleagues suggested, and we also read up about it, that let's work on zebrafish. So zebrafish is also a very specific, very well validated animal model for biological research. A lot of drug discovery happens on zebrafish. A lot of basic biology academic research happens on zebrafish, where a lot of things have been now deciphered through those experiments. So it's an acceptable model for biological research. It's an vertebrate, and it has pretty much a similar kind of pathophysiology or the normal physiology as a normal vertebrate would have. But for our kind of experiment, we had one more challenge. We were looking for the lungs, and fish, unfortunately, have no lungs. So what are you going to do? That becomes one more challenge. So then we thought that, does fish has anything where there's any air is stored? And the answer is yes. There's something called air sac, something called swim bladder, what you see over here. This is a swim bladder of a fish, which has anterior and posterior lobes. This swim bladder is used by the fish for remaining floating in the water. To go up and down, this volume of a swim bladder air is used by the fish. So this is actually air sac inside a fish. Now, that is not being used by the fish for gaseous exchange, which is a function of the lungs in our case. So what we do again? So then we went ahead and took our human alveolar epithelial cells, what we were working earlier, A549 cells, and injected those cells in the swim bladder of the fish, which you see over here, and see whether these cells, what you see here, get any homing in a normal bladder like this. So this is a normal bladder where is no cells have been injected into. While here, what you see in each circle, these are the human lung epithelial cells sticking very well there. And they made a new home for themselves there. It took them 7 to 8 days to really attach and proliferate. And they start to make colonies right inside that fish swim bladder. So we were successful to get an almost humanized zebrafish, which has a live living colony of human lung cells inside them. But that was not good enough. We have to do something with the SARS-CoV-2. So we got SARS-CoV-2 spike protein, what we were using earlier in our in vitro experiment, and injected right into those areas, right over here, and then measure the different phenotype that fish has to go through. And that becomes our first animal model, a transgenic, not transgenic, xenotransplant humanized model of zebrafish, where we could actually see the effect induced by SARS-CoV-2 spike protein. And the effects were hemorrhage, which also happens in the human thing. We could also measure the fever. Yes, we could measure the fever. I'll explain to you how would we measure the fever, and could also measure the differential infiltration of various inflammatory cells in those swim bladders. We could see a differential temporal infiltration of granulocyte, macrophages, and lymphocytes right there where the SARS-CoV-2 spike protein have been injected. So we felt now we have something in our hand. Literally, my team was happy to say, yes, we have new toys where we can play around with the condition we are trying to see and see if there is a very specific effect of the drugs that we will be working at. We were having lots of different formulations, single plant herbs, single plant extracts, Coronil, Swasaribati. There are several other things that we worked upon, but we didn't have a right model to play around. This was one of the early successes we got. This was, we actually finished in the May 2020 and went ahead to start doing the detailed experiment.
Dr. Rahul, forgive me. I think we have to now sort of time is a constraint here. I think you have another 7 to 8 minutes. I'm sorry.
I will go very quickly, sir. Thank you very much, sir. Thank you very much. I'll go very quickly.
Thank you.
So this is the fever that I was talking about. We could smartly measure the fever of the fish by looking at the specific temperature that they want to stay. So fish are actually they are the cold-blooded animals. They do not change their body temperature. Their body temperature gets changed from their external surrounding, unlike us. But we then got a very fancy different chamber thing where we kept different temperatures of the water and see the amount of time spent by the fish at differential temperature, and that becomes an indication of the temperature fish would be staying, and that would actually call behavioral fever. So the animals which were given SARS-CoV-2 spike protein, they spend more time at 37 degrees Celsius temperature box, and once they were treated with Coronil, they tend to come back towards their normal temperature, which is 29 degrees Celsius. And we could see that, yes, that way the behavior of the fish could be monitored. Next slide shows the spike protein induced inflammation in that area. These are the data from there. And we also measure cytokines in the fish as we were measuring in the our in vitro experiment, and we could see, yes, IL-6, TNF-alpha, and IL-10 was significantly downregulated when we treated the fish with Coronil. The data is there on the right-hand side. This is at the 4th day, this is on the 7th day. Next slide shows the critical component, and I want 2 minutes to spend here, the randomized placebo-controlled clinical trial on human positive patients which were positive for COVID-19. Here, we took 5 compounds in it, basically ashwagandha, which is Vidania somnifera, Tinospora cardifolia, and Ocimum sanctum, along with Swasaribati and Anutela. So all of those 5 components were actually given to the patients who were suffering or who were COVID positive at the time of entering into the trial. This trial has been conducted at the NIMS University, Jaipur, under the leadership of Professor B.S. Tomar. This trial has been registered in CTRI and has also been enlisted in the WHO Clinical Trial Registry. I will take you very quickly through the outcome of this work. This is the primary endpoint of the trial, the number of people becoming negative, positive becoming negative, and number of days it takes. So we also had a placebo group where the placebo tablets of these drugs was given, and this is the treatment group. At the 7th day, 100% people in the treatment group shows a recovery, where the placebo groups has only 60%. So that would mean that the treatment has a beneficial effect of the treatment benefit on the patients. This is a similar kind of data that you see. This finding, along with the next I'm going to show, has been published in Phytomedicine last month. So most of this information there on this reference, if you really want to go more in detail, that you can go over there or we can discuss as well. In human patient, we also look for hsCRP level, which are the classical marker for inflammation, highly sensitive C-reactive protein. This is the placebo group, this is the treatment group, and if you look at the changes, there's 12 times lesser amount of CRP present in the treatment group. We measured TNF-alpha, similarly what we saw in our in vitro experiment. This is the placebo group, this is the treatment group, and if you see the percentage change, 20 times lesser TNF-alpha present in the treatment group. And we also had clinical observational studies done separately in other COVID care center, where we found better pharmacological effect in the people who were taking these Ayurvedic medicines. I'll go very quickly from here. And in addition, we wanted to measure the psychological impact and the quality of life of the patients who were suffering from COVID-19 infection. And if the Coronil kit or Coronil tablet available to them, does it affect their psychology too? So we had a specific team that does this assessment. Close to 367 participants was enrolled in this survey, and we could find that increased treatment satisfaction with the Coronil kit lead to a decreased psychological impact and increased quality of life in study participant. That means it does have an effect on the psyche of the people and the social peace. This work is already under final review at Journal of Clinical Medicine. So summary so far from this work that we have done is as follows. The pharmacological effect of Coronil as an inhibitor of SARS-CoV-2 spike protein mediated entry in the human cells have been established, that I showed to you. And I hope I could convince that really happens. Coronil controls the cytokine storm. It also affects the interaction between spike protein and human ACE2 receptor. Coronil affects the pathological symptoms measured in humanized zebrafish. We saw in clinical studies that there is a faster recovery in the people who took the components of the Coronil. In psychosomatic study, the Coronil treatment significantly reduced the anxiety and depression and increased the quality of life of the patients who were COVID positive was taking Coronil tablets. We also did an additional experiment, which I didn't talk to you, is doing a tox study. So we conducted a GLP tox in rats and rabbits for 28 days, up to 1,000 milligram per kg per day. And we found not a single toxicological parameters that got modulated by this treatment. So we were very happy to actually observe a very clean tox profile of Coronil in rats and rabbits for 28 days, 1,000 milligram per kg under GLP. So I also want to show this because this was there in quite a while in the media, that just to show that yes, Coronil was also awarded a COPP WHO GMP certificate by the Drug Controller General of India. This is certificate of pharmaceutical product as per the guidelines set by WHO under good manufacturing practices. This is the copy of the certificate that we got from Government of India. And on the right hand side, what you see, the world map, okay, with this certificate, now Coronil can be exported to 158 countries of the world, which is the setup that CISSA has been set up with the help of Drug Control General of India.
Forgive me, Dr. Varshney. I think we have to conclude now.
This is the last slide, sir. Yeah. So this is the manufacturing license for the Coronil, which have been given by the state licensing authority. And this is the list of, this is the listing on WHO website for enlisting the clinical trial that we conducted with the component of Coronil. So thank you very much indeed. I really appreciate your time and your attention for the presentation that I have. This, I just leave you with the all the work we have done on in the form of a timeline, that what we did and what time we really went ahead with that. So thank you very much indeed. I really appreciate that.